Okay, so welcome back to this next video in which we are discussing voltage-gated ion channels. Okay, so far what we've been discussing is voltage-gated potassium channels, and we've also discussed the closely related family of calcium-gated potassium channels. What we now want to turn our attention onto is discussing another family of voltage-gated ion channels, which is the voltage-gated sodium channels. Okay, so we're going to start off by discussing the structure of voltage-gated sodium channels, or rather we're just going to initially discuss the actual pore portion of a voltage-gated sodium channel, so the absolutely essential portion of a voltage-gated sodium channel. We'll leave the accessory subunits out for now. Okay, then what we're going to talk about is the gating mechanism, so how a depolarization in the electrical potential difference across the cell membrane can lead to the opening of a voltage-gated sodium channel. And we'll see that it's exactly the same as what we've seen for voltage-gated potassium channels. Then we'll talk about the permeation and selectivity mechanism, and that suddenly is different. Okay, that's where we'll have to wake up and concentrate harder. Okay, because the permeation and selectivity mechanism is completely different from what you might expect. Okay, if you were going to create a permeation and selectivity mechanism for voltage-gated sodium channels, then you might imagine, let's use the success of what we've had in voltage-gated potassium channels and just narrow the selectivity filter down a bit. Okay, the reason sodium couldn't go through the voltage-gated potassium channel selectivity filter was because it was too small. So just make those coordination sites smaller. Okay, make the selectivity filter narrower might be your response. Okay, so that sodium can give up its hydration shell in an energetically favourable way and make its way through the selectivity filter, and yet the selectivity filter will be too small, too narrow for potassium to move through. Okay, however, that's not what happens. You actually make the selectivity filter wider, okay, and instead what you let happen is you let the sodium ion go through with some of its hydration shell still attached, so it only gives up part of its hydration shell. Okay, and we'll just look at that in much more detail later on. Okay, right. Then after we've done the permeation and selectivity mechanisms, we'll talk about inactivation. Okay, again we have a fast inactivation mechanism or an N-type inactivation, and we have a slow type inactivation mechanism or a C-type inactivation mechanism. Then finally what we'll turn our attention to is the beta subunits, the accessory subunits of voltage-gated sodium channels. And then actually, I forgot one bit. The real final bit is going to be, we'll discuss some examples of neurotoxins which can block voltage-gated sodium channels, including tetrodotoxin from pufferfish and saxitoxin from marine plankton. Okay, right, so let's start off by discussing the structure then of voltage-gated sodium channels. So VGNCs will be my abbreviation for voltage-gated sodium channels. So we're going to start off by discussing the essential part of the voltage-gated sodium channel structure. Okay, and again we're going to start off with a basic picture, and then we're going to build it up to the more true picture of what these things look like. Okay, but that's a more complicated picture. And indeed what they look like is very similar to what voltage-gated potassium channels look like, with one huge twist. Okay, and I'll give you that huge twist now. So let me draw a very basic picture then of the essential pore portion of a voltage-gated sodium channel. So here it is in cylinder form, sitting in the cell membrane like so. So this is our voltage-gated sodium channel. And here comes the big twist, the big difference between voltage-gated sodium channels and voltage-gated potassium channels. If this was a voltage-gated potassium channel, I would now be about to split that into 4 separate proteins. I would now be doing the speech about how it was a tetramer and how it was 4 separate proteins stuck together. In the case of voltage-gated sodium channels, I don't need to do that speech. The pore-forming unit of a voltage-gated sodium channel is one enormous great protein. Okay, one protein with an N-terminus and a C-terminus. It's one polypeptide called the alpha subunit of a voltage-gated sodium channel. Okay, So this is one protein. That's all you need to make a voltage-gated sodium channel. You don't need any joining together of more than one polypeptide. It is one polypeptide. It can fold into the entire pore-forming domain on its own, basically, okay? And it's functional as a voltage-gated sodium channel on its own. Okay, however, what people do is they divide voltage-gated sodium channel alpha subunits into 4 separate domains, which are analogous to the 4 separate subunits of a voltage-gated potassium channel. So we are still going to divide this up, but I'm going to do it with dashed lines rather than solid lines. Okay, so here I'm dividing it up with dashed lines, basically, or dotted lines. Okay, and these are the 4 separate domains of the alpha subunit that are analogous to the 4 separate subunits of a voltage-gated potassium channel. Okay, and we'll call this one here domain 1. Okay, we'll call this one back here. Okay, so this one here, I might even color code them in. So we'll have domain 1 here in red. Okay, and we'll have domain 2 back there, the second quarter, in blue. Okay, we'll have domain 3, that one back there, in green. Okay, so that's color coded in green, and then we'll have domain 4 right at the front here, like so, and we'll have that one in turquoise. Okay, so what I now want to do is show you the actual membrane spanning topology of an alpha subunit of a voltage-gated sodium channel, show you the 4 separate domains, make you appreciate how basically an alpha subunit of a voltage-gated sodium channel is just like 4 subunits of voltage-gated potassium channels joined together, and then I'll give you a more true picture of what this actually looks like, which will make it look a lot more like the voltage-gated potassium channels. In fact, it will look identical to the voltage-gated potassium channels. Okay, so this is a very crude picture, just to get this central concept across, that it's a single protein with these 4 separate domains that makes up a quarter of the voltage-gated sodium channel alpha subunit each. Okay, right, so let me now show you the membrane spanning topology then of an alpha subunit of a voltage-gated sodium channel then. Okay, so once again, here is the cell membrane. I'm going to need a lot of cell membrane for this because this is a big protein. Okay, and it's going to be all one protein. Now, the N and the C termini, they both start on the cytoplasmic side of the membrane. So we'll say that the bottom half is the cytoplasmic side and the upper half is the extracellular fluid side, as always. Okay, so here we'll start off with the amino terminus then of our alpha subunit of our voltage-gated sodium channel. And then what's going to happen is the polypeptide is going to span the membrane once, span the membrane a second time, a third time, a fourth time, a fifth time, you'll have a p-loop, and then a 6th time, just like a subunit of a voltage-gated potassium channel. Okay, however, it's not going to stop there. It's going to repeat the same thing 3 more times. Okay, so now I'm going to repeat that entire thing again, and then again. Okay, so here we go again. And then one more time, just for luck. Okay, so a 4th time here. And then you'll have the C-terminus here. So that is the membrane spanning topology of an alpha subunit of a voltage-gated sodium channel. It's like 4 alpha subunits of voltage-gated potassium channels joined together. Okay, right. Now let me talk about these 4 different domains then. Well, you can probably guess what the 4 different domains are then. Okay, so this domain here, this is domain number 1. Okay, so the first one of these repeated structures that is analogous to a alpha subunit of a voltage-gated potassium channel is called domain 1. Then the second repeat here is called domain 2. Okay, in blue there. The third repeat here is called domain 3. Okay, and then the 4th repeat over here, which we'll have in turquoise, is called domain 4 here. Okay, right, so those are the 4 separate domains of an alpha subunit of a voltage-gated sodium channel, and they each make up a quarter of the voltage-gated sodium channel. Some more terminology then. Okay, so these linkers that we have between the 4 different domains, we have 3 of them. We have a linker here, a linker here, and a linker here. These have special names. So the linker between domain 1 and domain 2, that's called the 1,2 linker. Okay, and we'll be using this terminology later on, which is why I'm giving it to you. Okay, so this is called the 1,2 linker here, and I'll just colour that in pencil there. So that's the 1,2 linker. This one then you can probably guess its name. It's called the 2,3 linker. Okay, here. And then I'll just colour that one in purple as well. Okay, and then finally, the final one here is called the 3,4 linker, and we'll be seeing something very important about the 3,4 linker later on. It's actually going to take the place of the inactivation ball for voltage-gated potassium channels in the fast type inactivation. Okay, and that's why generally, with regards to fast type inactivation in voltage-gated sodium channels, you will call it fast type inactivation rather than N-type inactivation, because in the case of voltage-gated sodium channels, it's mediated by a portion that's not really near the N-terminus at all. Okay, so fast type inactivation is called N-type inactivation because it was first understood in the voltage-gated potassium channels, and there obviously it involved very much so the N-termini of the alpha subunits of the voltage-gated potassium channels. However, you have the analogous thing in voltage-gated sodium channels, but it involves this 3,4 linker, which is nowhere near the N-terminus. So you can call it N-type inactivation in voltage-gated sodium channels simply because that's what it's called in voltage-gated potassium channels, but it's probably better to call it fast type inactivation. Okay, right. So a final piece of terminology I'm going to go through before we actually go on to talking about how these things are really arranged into a structure, okay, is I'm going to tell you how you name up the individual alpha helices then. So we know that in the alpha subunits of voltage-gated potassium channels, the helices here are labelled S1, S2, S3, S4, S5, S6. So how do you name them up in this alpha subunit of the voltage-gated sodium channel? Do you call this one S1, S2, S3, S4, S5, S6, and then go on S7, S8, S9? No, you don't. The way you do it is you just tell the person which domain you're talking about, and then you name the alpha helix as it would be named in a voltage-gated potassium channel alpha subunit. So for instance, let me give you an example. If I wanted to name up this one here, that now highlighted in orange, you would start by giving what domain it's in. So you put a D for domain, and then you say it's domain 3. Okay, and if you like, you can put it in Roman numerals. If you're less pretentious, you can put it in Arabic numbers. Okay, domain 3, and then you'd say which alpha helix is it in domain 3, so you'd then put S2. Okay, and that looks really odd with the Roman numeral there, so I'll replace it with an Arabic numeral, D3, S2, like so. Okay, so that's how you label up all the alpha helices of this structure. You'd say which domain is it, and then which alpha helix is it, if it was just an individual alpha subunit of a voltage-gated potassium channel, rather than a domain within a alpha subunit of a voltage-gated sodium channel. Okay, right. So that then is the terminology done. What I'm going to tell you about just before I tell you about how these things are actually arranged in real life, okay, the real picture of how they're arranged, okay, and it's going to be exactly what we've seen for voltage-gated potassium channels, okay, with the twist that they're all connected together, okay. Let me tell you about the fact that there is more than one different type of alpha subunit of voltage-gated sodium channels. So we saw this with alpha subunits of voltage-gated potassium channels. There were 40 different alpha subunits of voltage-gated potassium channels. There is not just one alpha subunit of a voltage-gated sodium channel. It would be beautifully simple if there was, but it's not that way. However, it's not that bad. There are only 9 different alpha subunits of voltage-gated sodium channels in humans, and again they have a beautifully simple naming system. So the 9 different alpha subunits of voltage-gated sodium channels are called, whoops, not like that, the NAV1s, and then the first one is called NAV1.1. Okay, so NAV1.1 is a gene for a alpha subunit of a voltage-gated sodium channel. Then it goes on, you have NAV1.2, NAV1.3, and I said you had 9 of the things in total, so it goes on all the way up to NAV1.9. So that's the naming of the 9 different genes for the alpha subunits of voltage-gated sodium channels. Okay, and they will all have the same basic membrane spanning topology. Of course, they will all code for proteins that will have different sequences of amino acids, but their grander structure is the same, basically. They all have the same membrane spanning topology. So what I've discussed here applies for all 9 of them, basically. Okay, so just to tell you about the expression profiles of these, okay, so most of them are found in nervous tissue, so most of them are found in neurons. Some ones that are notable because they're not found in neurons are NAV1.4 and NAV1.5, okay, so all of the other ones apart from NAV1.4 and NAV1.5 are principally found in neurons of the peripheral and central nervous system. Okay, but NAV1.4 is the type of alpha subunit of voltage-gated sodium channels that is found in skeletal muscle, and I thought I'd completed it just by putting muscle there, but of course I haven't, because this one's found in muscle as well, just a different type of muscle, so I need to put skeletal muscle there. So NAV1.4 is the alpha subunit of voltage-gated sodium channels that is principally expressed in skeletal muscle tissue. Okay, another type of excitable tissue capable of firing action potentials that isn't obviously nervous tissue. NAV1.5 then, this again is principally found in a tissue other than nervous tissue, and it's the other type of excitable muscle tissue, cardiac muscle tissue. Okay, so NAV1.5 is found in cardiac muscle tissue principally. Okay, so all of the others, NAV1.1, NAV1.2, NAV1.3, NAV1.6, NAV1.7, NAV1.8, and NAV1.9, they are all found in peripheral and central neurons, basically, whereas those two are the ones that are found in skeletal and cardiac muscle cells and are responsible for action potentials in skeletal and cardiac muscle cells. Okay, right. So those are the different types of alpha subunits of voltage-gated sodium channels. As I say, these basic structural features that we're talking about apply for all 9 of them, and indeed everything that I say from now on about how this actually folds up into a 3-dimensional shape, and how they are voltage-gated, and how they allow sodium ions to move through them, and how they inactivate. All of that applies for all 9 of these alpha subunits. Okay, so I'm not now going to dwell on the fact that there are 9 different alpha subunits in the way that I did dwell on the different alpha subunits for the calcium-gated potassium channels. Okay, because everything we say is now going to apply for all 9 of these, whereas obviously things were different for the different types of calcium-gated potassium channels. Okay, right, so I'm now going to go through with you how this actually folds up into a 3-dimensional shape. Okay, how is this actually organised? Because it's more complicated than all of the domain 1 just being crunched into one corner. Okay, and it ends up looking very much so like our voltage-gated potassium channel. Okay, right, so what I'm going to do is once again draw this from above. Okay, so you're going to have the pore domain actually in the center, and then again you're going to have these voltage sensing domains sticking out from the sides basically, and it's the same portions of each domain which make the pore and the voltage sensing domains as you would expect if they were individual subunits of a voltage gated potassium channel. So once again, S1 to S4 for each of these domains is going to be making the voltage sensing domain, okay, whilst S5, P and S6 are going to be making the pore forming domain. So from each of these four domains, S1 to S4 is going to be forming a voltage sensing domain, okay, and again from each of these four domains, S5, P, S6 portion is going to be contributing to the pore basically. Okay, right, so let me now try and draw the voltage sensing domains on this picture here. Okay, and once again we'll colour code this up, okay, and I'll just tell you the colour coding in a moment. So here are the four voltage sensing domains. Right, so I'm now going to draw out a single domain and we'll colour code it in. Okay, so here is a single domain. I'm not saying which one it is, it will apply for all four of them. Okay, and now I just want to colour code things in. So we'll colour code S1 in yellow here. Okay, we'll colour code S2 in green. We'll colour code S4 in blue. We'll colour code, S3 in blue. We'll colour code S4 in orange here. Okay, we'll colour code the S4, S5 linker in red here. Okay, we'll colour code the S5 in turquoise here. We'll colour code the descending P-loop in vivid purple. We'll colour code the ascending P-loop in yellow. And then finally we'll have to use red again. We'll colour in S6 here in red again. Okay, right, so now what I want to show you is the arrangement of these helices in the actual tetramer, and I want to dwell more on these links between them, because basically what we are going to see is exactly the same as what we've seen for voltage gated potassium channels. The reason I'm doing this again is so that I can add on to this picture the linkers basically, so that I can make it more obvious. So let's start off by putting in domain 1 then. Okay, so I've said that we're going to have domain 1 in this position here. Okay, so its pore forming domain is therefore going to be this portion here, but its voltage sensing domain, as always, is going to be over here. Okay, so let's start by putting in its voltage sensing domain. So here is the S1 helix for domain number 1 here. Okay. Then you'll have the S2 helix here in green. Again, the arrangement is exactly as it was for the voltage sensing domain in the alpha subunits of voltage gated potassium channels. Here then is the S3 helix in blue, and here is the S4 helix in orange. Now, let me just put something in here, because I'm now going to actually demarcate the fact that that is where the N-terminus of this massive great alpha subunit has actually begun. Okay, so on the cytoplasmic side of the membrane, the N-terminus begun with the S1 helix here of domain 1, then it went into S2, then it looped back up into S3, looped over into S4, and now I'll put on the pore forming domain. Okay, so once again, S5 is going to be an outer portion, okay, and S4 is going to be linked to S5 by the S4, S5 linker here, okay. Then, after we've had S5, which will be coming from the cytoplasm up to the outside, you'll then have the P-loop. Here's the descending P-loop. Then you'll have the ascending P-loop, which will be the portion that actually lies the pore there, in yellow. Okay, and then, after you've had the ascending P-loop, you'll then loop over into the S6 alpha helix, which will go down, and it will be sort of behind the ascending P-loop there. Okay, so in red. So the key thing that we've done this for is now so that we can see how we're going to have these linkers. So we now want to understand the 1,2 linker now. Okay, so the 1,2 linker is going to take us from the S6 helix of domain 1 here, into the S1 helix of S domain 2 here. Okay, so let's just put on the S1 helix of domain 2. Here is the S1 helix of domain 2. So basically, on the cytoplasmic aspect, you're going to have a linker here from the S6 of domain 1 to the S1 of domain 2, basically, and I'll colour that linker in vivid purple. Okay, and then we just repeat this on. So, after S1 of domain 2, you'll then have S2 here, here is S3, and here is S4. Okay, and then you can just continue this on, basically. You'll now have the S4, S5 linker from S4 here to S5, which will be back here. Okay, so now I'll put in S5. Okay, then after S5 has come up through the membrane to the outer aspect, you'll then have the P-loop. Here's the descending P-loop of domain 2 here. Then we'll have the ascending P-loop of domain 2 lining the pore there. Okay, and then behind the ascending P-loop of domain 2, you'll then have the S6 helix, which will go down into the cytoplasm once again. And there is now domain 2 finished, and then again we'll have another one of these linkers which will link us from the S6 helix to the S1 helix of domain number 3. And you can imagine how this can continue on. I'm now just going to finish this picture off, and then we'll discuss the full thing. Right, so let's now mark on the voltage sensing domains for the two remaining domains here. Okay, so we'll start by putting on the S1 helices here in yellow. Then let's mark on the S2 helices for these two remaining domains, domain 3 and domain 4, there in green. Okay, then we'll put on the S3 helices here in blue. Okay, then the S4 helices here in orange. Okay, then we'll have the S5 helices here in turquoise. Then we'll have the descending P-loops here in vivid purple. Okay, we'll then have the ascending P-loops, which are lining the pore right at the centre there in yellow. Okay, and then finally we'll finish off with the S6 helices here in red. Now let me just add on the S4, S5 linkers. So here is the S4, S5 linker for domain 3 here. Here's its voltage sensing domain linked to its pore forming domain here. And here then is the S4, S5 linker for domain 4 here. Here's its voltage sensing domain. Here is its pore forming domain. Okay, and the final thing that we really want to put on here is these linkers. So here is domain 1 here. Here's its voltage sensing domain, pore forming domain. Here is domain 2 and the domain 1, 2 linker here. Here's domain 2's voltage sensing domain. Here's its pore forming domain. Okay, now what we want is the linker between domain 2. Here is the S6 helix of domain 2. Okay, and we want the linker from the S6 helix of domain 2 to the S1 helix of domain 3. Okay, so I'll put that in there. So there is the 2,3 linker there, and of course it will be on the cytoplasmic side, but allow me that incorrect feature here, just for conceptual purposes. Okay, here then is the voltage sensing domain of domain 3. Here's the pore forming domain of domain 3, and then we now want the linker from the S6 helix of domain 3 to the S1 helix of domain 4. So let's now put that on here. So there now is the 3,4 linker, which I've said is going to be really important. Okay, then we've got the voltage sensing domain of domain 4 here, and the pore forming domain of domain 4 here, and then it ends. Here is the S6 helix of domain 4, and on the cytoplasmic aspect you'll then have the C-terminus of this alpha subunit. Okay, so that then is how these alpha subunits of voltage-gated sodium channels are believed to actually fold up. Okay, and I should say that we don't yet, to my knowledge, have crystal structures of human alpha subunits of voltage-gated sodium channels, or even mammalian alpha subunits of voltage-gated sodium channels. Okay, so this is our best guess of what it is. We have very good evidence that this is what it is, okay, but we don't fundamentally have those crystal structures yet, so we don't absolutely know that this is the way it is. Okay, right. So, now that I've shown you that, it should be very obvious what the gating mechanism is going to be. Okay, how analogous it is to voltage-gated potassium channels should scream at you that the gating mechanism is going to be analogous to voltage-gated potassium channels, and indeed it is analogous to voltage-gated potassium channels. Once again, each of the S4 helices of these 4 voltage sensing domains that we have is positively charged. It has lots of arginines and lysines within it, which end up being protonated at physiological pHs. Okay, so when the electrical potential difference across the cell membrane is depolarized, the amount that these S4 helices are being pulled inwards towards the intracellular compartment is going to be hugely reduced, okay, and that's going to cause the voltage sensing domains to all move out a little bit, okay, and that will yank on the S4, S5 helices and then pull the S6 helices out of the intracellular aspect of the internal pore. Again, I should have probably said before I discussed the gating mechanism that the V-gate is believed to be the same. Okay, so the reason that the channel is usually closed at resting membrane potential of negative 65 millivolts is once again believed to be that the S6 helices at the intracellular aspect all converge in on the center of the internal pore basically and occlude the movement of potassium ions through the internal pore. Okay, and that's once again called the V-gate. Okay, right, so if we want to open the voltage-gated sodium channel when we depolarize the electrical potential difference across the cell membrane, we need to open the V-gate, we need to pull these S6 helices outwards, and again it's believed to be done by the same mechanism of the voltage sensing domains moving outwards, okay, moving upwards, and then pulling on the S4, S5 linker, and that causes the S6 helices to all move outwards away from the center of the pore, opening up the internal aspect of the pore, and now sodium ions are going to be able to move through that pore, at least transiently before inactivation occurs. Okay, right, so we'll call it there for this video, and then in the next video what we'll talk about is firstly the permeation mechanism, which is really different from the permeation mechanism in voltage-gated potassium channels. Okay, we'll then talk about inactivation, which is very much so analogous but different fundamentally from what we've seen in voltage-gated potassium channels. And then what we'll talk about is the accessory subunits, the beta subunits, and then the neurotoxins.